Good morning. I'm Jeff Eisenberg, CEO of Xenetic Biosciences Inc. I appreciate your interest in the Xenetic story this morning, and I'd like to thank Noble for inviting Xenetic to participate in this conference. I will be making forward-looking statements today, so please consider what I say in light of the most up-to-date information in our SEC filings and on our website. So the story of Xenetic is really at this point a CAR-T story. And the story revolves around the proprietary technology that we have called XCART, which is a personalized CAR-T platform which targets cancers with a patient and tumor specific approach. Now that differs from, and I'll get into this in the presentation, from other currently commercialized CAR-T therapies, because we target tumor specific antigens that are independent of CD19 or other antigens common to all B cells. We are preclinical stage, advancing toward a phase 1 study, and our lead program is in the large and growing NHL blood cancer market. We're advancing our technology through the use of 2 academic collaborations, which I'll also discuss. And we feel in a lot of ways that while we're building on the proven success of CAR-T therapy, we have a highly differentiated approach, which should stand out in the field. We also believe that with the unique technology we have in the CAR-T space, there is an opportunity for us to create significant value even during relatively early stages of development. Now, there is a second technology in the company as well, which we call Polycylic. It's a biologic half-life extension platform. And a couple of points to make on Polycylic initially. Number one, we have an existing exclusive license agreement with Takeda. That's in the field of blood coagulation factors. And under that agreement, we are receiving an ongoing royalty stream, and of course, reporting those royalties on a quarterly basis in our SEC filings. And as well, we recently announced successful phase 3 study results from our partner PharmSynthese for an EPO product employing the Polycylic technology in Russia. I'll talk about that a little bit more as well. This is a look at our senior management team. It's a core group of senior leaders with significant experience in pharma and biotech. Each of us well over 20 years in public and private companies of various types. We are a small team. We've intentionally kept our overhead small and done a lot of our work through the use of external advisors and consultants. And some of those are shown on this slide, which is our scientific advisory group. Now, this group is really put together for the XCART technology and to help us advance the XCART platform. It's comprised of experts in lymphoma and cell therapy, including the head of the clinical CAR-T program at Harvard, the head of blood and marrow transplant and hematologic oncology at Miami Cancer Institute, who had also spent 20 plus years at MSK, as well as CAR-T experts out of Baylor, helping us with all aspects of CAR-T design, clinical design, and clinical and commercial use cases. We're also working with some of the founders of the technology, and I'll talk about that as well as experts in CMC and regulatory. So let's dive into the XCART platform. As I mentioned, we are focused in the area of lymphoma initially, and in particular, we're going to hone in on the area of B-cell lymphoma. Of course, B cells, the function of B cells is to produce and secrete antibodies in the form of B cell receptors, which then selectively target a given antigen. And we'll build on the importance of that B cell story as I talk about how the XCART technology actually works. Now, the NHL market, as I mentioned, is a large one and a growing one. It's about 77,000 new cases annually. It does represent 90% of all lymphomas, NHL does. And about 90% of all lymphomas are non-Hodgkin lymphoma of the B-cell variety. So let's take a look now a little bit of cellular biology and how that plays into tumor formation and growth. So if you look on the left side of the slide, this is a representation of normal circulating B cells. And as you can see, each of these B cells expresses the antigen CD19 on the cell surface. Each of these B cells also expresses a unique B cell receptor, which is depicted here by the different colors on the surface of these B cells. Now again, the current commercial therapies only target the CD19 receptor, which is common and present on all B cells. Now if you look on the right, we can talk about tumor development. So each healthy human has billions of these unique B cell clones in their body. When one of them transforms and becomes malignant, that malignant B cell retains its unique BCR. And then when that malignant B cell begins to proliferate and form a tumor, each members of that clonal population, the tumoral clonal population, also retain that unique BCR, which then forms our target. So because B cells express both CD19 as well as this unique BCR, in a sense there's a choice of how to target and how to treat them employing a target of the CD19, a common antigen, or in our case the B cell receptor. So clearly the anti-CD19 targeting CAR T therapies work. There are 2 or actually 3 now approvals with Yescarta, Kymriah, and Tecartus. Clinical data showed significant efficacy with very high complete or partial response rates, remission rates, survival rates. Obviously at the time these were first reported, especially these were groundbreaking in these previously very serious cases of late-stage lymphoma. And again, if you look on the right, you can start to see that while these therapies work, there is a challenge with the way they work. Because if you introduce an anti-CD19 targeting T cell, which is depicted on the gray box in the middle, it is going to bind to the CD19 receptor both on normal B cells as well as on malignant B cells, and thereby kill and eliminate all B cells. Now, partly because of that, but also just because of the design of the early CAR T products, we know there are significant shortcomings associated with these currently approved products and definitely a need for improvement. So most of you will be aware of toxicity problems like cytokine release syndrome and neurotoxicity, which are recognized for CAR T products in general. And for CD19 targeting CAR T cells, as I noted, B cell aplasia is a natural consequence of treatment since the CD19 is a surface antigen that's present on both normal and tumor B cells alike. On the other hand, tumor cells with low or no surface expression of CD19 lack sufficient target to be recognized and killed by the anti-CD19 CAR T cells, which leads to insufficient initial response to treatment in some patients. Furthermore, long-term durability of response to CAR T can be influenced by either exhaustion of the CAR T cells themselves or by the emergence of tumor cell variants that have escaped the CD19 targeting mechanism. Now, this so-called CD19 escape accounts for a significant fraction of the relapses from anti-CD19 CAR T therapy. The next slide, I'll get a little deeper into the CD19 escape phenomenon. So again, let's dig a little deeper. Now, as shown on the right, under selective pressure from anti-CD19 cell killing, mutated tumor cells can arise, which again either lack surface CD19 entirely or display mutated CD19s that are no longer recognized by the CAR T targeting domains. In either of those situations, the phenomenon of CD19 escape can lead to CAR T relapse. And in fact, it's estimated that CD19 escape accounts for up to half of all CD19 CAR T relapses and up to half of CAR T treated patients to date, according to some reported studies, will suffer relapse within 12 months. An important point for us and for XCART is that experts we've consulted with expect that this incidence of patients who experience CD19 escape or who have CD19 negative tumors will increase in the future as more CD19 targeting therapies are employed clinically. So that definitely presents an opportunity for XCART and really reinforces the XCART proposition with respect to addressing CD19 escape. And this slide goes into a kind of a step back look at why and how this is an advantage of XCART. So remember, XCART targets the B cell receptor. So if you look on the left side of this left part of this slide, it shows that XCART can target a tumor B cell similarly to CD19 CAR T cell as long as the CD19 antigen is active. So if a CD19 antigen is normal expression, no problem. But on the right, only the XCART can target a B cell with a mutated CD19 or an absence of CD19 altogether. So we certainly believe that the CD19 negative population represents a real unmet need and a differentiating advantage for XCART. But I don't want to lose sight of the fact that XCART targets only the malignant B cells, which is its kind of principal inherent advantage. So on this slide, if you start looking at the left, again, the unique B cell receptor presents a tumor-specific target. So when you go to the middle, what you can see is that unlike the anti-CD19 CAR T cell, which I showed you will bind to and kill both the target malignant cells, but also the healthy B cells, the XCART, since it's not targeting a common surface antigen, it's targeting the B cell receptor, will only bind to and kill the malignant B cells, meaning we spare the patients from B cell aplasia, we spare the normal healthy B cells to continue to serve their function in the immune system. Now, this is some of the data that was presented in a scientific paper in Science Advances. And here what you're looking at is an in vitro experiment, a model cell line experiment, where model B cells were created and engineered to express the B cell receptor from one of a specific follicular lymphoma patient. And then model T cells were created and engineered to target specific BCR from one patient. And if you look at the lower left panel of this slide, you can see the results for FL patient 1. This is again the patient whose BCR was used to create the model B cell Raji FL1. And what you can see here is that the anti-CD19 CAR T model cell, which is shown in black, was effective in killing the B cells from patient FL1, which is what you'd expect. And the FL1 CAR T, or the CAR T cell that was engineered to target the BCR from patient FL1, also effectively killed those B cells. And importantly, the CAR T cell that was engineered to target the BCR from patient FL3 had no impact on patient 1. And that's exactly the result that was anticipated and desired to be seen and shown across the other two panels. So reinforcing the selective targeting mechanism. Now, there's clearly, as I noted, an unmet need in B-cell NHL. Here are just two representative subtypes of B-cell NHL, follicular lymphoma, which is a more slow-growing indolent cancer and a lower incidence level, and then diffuse large B-cell lymphoma, which is the largest population of B-cell NHL and a much more aggressive cancer. In each case, we believe that XCART can be a viable treatment option, as do our clinical experts. And I think it's important to note that there are clinical trials ongoing for the anti-CD19 CAR T therapies in some indications to try to move up the treatment line. So we would hope and expect that by the time XCART would be commercialized, that CAR T may be indicated for higher lines, probably second line at that point. Let me take a little bit of time and discuss the academic collaborations that I referenced earlier. So really what we were looking for entering 2020 was collaborations to help us move through preclinical development toward a phase 1 trial that had a couple of different attributes. Of course, we were looking for clinical investigators with, with experience and patients, treating patients with B-cell lymphoma. But we also wanted, and this was important to us, access to the capability for manufacturing infrastructure during the clinical phase. And we're able to do that with the two academic collaborations I'll discuss in a minute. With those up and running, here's a look at some upcoming potential milestones that we're working toward. So relatively shortly, we would expect to begin what we're calling an initial exploratory patient biopsy study in Eastern Europe. This will allow us to actually refine and exercise the XCART process on using actual patient biopsy material. So not a dosing trial, but a trial that allows us to use the actual patient material. Then we'll be looking to get some FDA feedback, seeking an INTERACT meeting. And then I think a key milestone that we're working toward is the commencement of the clinical manufacturing process development, which from our standpoint, will represent validation both internally as well as externally that we're moving closer toward the clinic. And again, the goal will be to commence a phase 1 trial in Eastern Europe and then ultimately to file a US IND. And so the two collaborations, the first is with Scripps Research, brings a tremendous benefit to us. Not only is it, of course, a leading research institution, very well known for its immunology expertise, but the scientists in the lab of Dr. Richard Lerner, who we've really been working with and who invented the XCART technology, are still our partners and are still the team that's helping us advance toward the clinic. And that includes things like designing and implementing the preclinical development program, optimizing the CAR-T design, cell engineering methodology, et cetera. And then through our strategic partner PharmSynthase, we were able to gain access to a number of academic institutions in Eastern Europe who will ultimately help us run the phase 1 trial for us. And during the preclinical phase will help us again optimize the overall XCART workflow and then develop those clinical manufacturing processes that we'll need to have in place before we can treat patients. So we feel like we've got a good team in place and a good group of partners helping us advance through preclinical development. Let me turn back just for a moment to Polycylic that I noted earlier and give a little more detail on that technology. Now again, it is a half-life extension platform technology, employs polycylic acid to modulate the PK and PD profiles of protein drugs. So you can think of it as a competitor to polyethylene glycol or PEG. It is clinically demonstrated to extend the half-life of therapeutic proteins, including the two referenced on the right, through our license arrangements. So I mentioned the one with Takeda. It's in the field of blood coagulation disorders. We are receiving royalties under that agreement, and that's really as a result of a third-party sublicense that Takeda entered into. It's those third-party sales that are driving the royalty stream. And in addition, our partner PharmSynthase recently announced positive pivotal phase 3 data for a polysilated EPO product. And that's used as a treatment for anemia in patients with chronic kidney disease. So we would expect, and we've been informed that our partner will be filing for approval in Russia and hoping to receive approval this year and hoping to receive approval. And that would also commence a royalty stream for us. We also think that these positive results validate the technology and do open up some additional partnering opportunities. As far as the financial snapshot, you can see our market capitalization. We were able to secure beef up our balance sheet a bit in the fourth quarter through a $6 million gross financing. That also had the effect of increasing our float and our average trading volume. Our cash balance shown here is a September 30 number. So when you add that to the proceeds of the financing, we were able to extend our runway and shore up our balance sheet. We had been saying we expected our cash balance to last through and beyond the middle of 2021. We think now with this additional cash, we should be comfortable into 2022. So in terms of a summary, we have a truly differentiated CAR-T technology with a unique targeting mechanism that we think really represents and solves potentially a clinical unmet need, targeting a large and growing market. While we're doing that, we have the existing license agreements with Polycylic technology, hopefully driving continued revenues and royalties. And so we really do think it's an exciting story, and we have a potential to have a transformative impact in the CAR-T space and really create significant value for shareholders. So that concludes my presentation. I'm now ready to take questions. And again, thank you for your time and interest this morning.
Thank you very much for your presentation, Jeffrey. So I'll start with the Q&A session, and I'll start with what is the rationale behind going after follicular lymphoma and DLBCL indications? Was there a preclinical data that drove the decision, or is it driven by the receptor that you found? So I'm just curious, what was the reason for the indication selections?
So 2 points. One, as I noted on that preclinical data that I ran through, in that particular case, there was follicular lymphoma patients whose biopsy material was used to screen and identify the BCR. But I tried to be clear that the follicular and DLBCL indications that were cited are representative examples of B-cell NHL subtypes. So we haven't decided yet which particular subtypes to target in our initial clinical studies. It's likely that this exploratory trial that I referred to will take a broad selection of B-cell NHL patients because we're really just looking to exercise the process. It's certainly likely that DLBCL will be a target because, again, it's a significant unmet need. It's the largest subtype, but we haven't yet committed to the whole range of subtypes that we're going to target.
Okay. And my follow-up question would be, does it mean you can go after many other indications, hematological malignancies, and does it depend on the receptor binding and other drivers, but the expansion of market strategy could go beyond what CAR-T is approved or what's beyond FL and DLBCL indications?
So it could go beyond FL and DLBCL indications within the B-cell lymphoma space. It really could target any B-cell mediated cancer. Beyond that would require some re-engineering, but the concept could be applied beyond B-cell lymphoma.
That's helpful. So one of the questions we receive is, what about targeting CD20, like biological rituximab treatments? So could you, I guess, use that receptor, or could you use it with combination?
Well, so there are clinical programs ongoing by other players in the field targeting CD20, as well as CD22 and in some cases dual targeting CD19 and CD22. Again, I would expect those to work, but in each of those cases, you still have the potential issue of antigen escape. And one of the things that we were obviously seeking to do was to select a target that nobody else was pursuing, and that's why the BCR we really think represents an elegant target in this area. In part because we certainly believe we have to show this clinically. We certainly believe it mitigates or eliminates the issue of antigen escape, as well as potentially, because of that, providing a valuable rescue therapy for CAR-T relapse patients. So even if clinician still decided to use, let's say, a CD19 or a CD22 targeted CAR-T for a patient initially, if that patient were to relapse because of a loss of their antigen, they're really at that point out of options. XCART can be a valuable rescue therapy in that situation.
That's helpful. So another question comes to mind is, I think commercial viability of CAR-Ts. There is the cost effectiveness is an important component. So what would you think about the pricing of your treatments and the commercial success, commercial viability of it, I think?
So I would expect that by the time we got to market, we would price comparably to what the competitive products would be priced at that time. And it's a little difficult to tell, obviously, because there's only still a couple approved. So I wouldn't expect that our selling proposition would be, a lower price. Our proposition is a unique target for all the reasons that I stated. So I think from a commercial standpoint, it's really the unique targeting that would be the selling proposition.
That's helpful. And another question I have, Jeffrey, is the Takeda royalties. Can you give us some information on the agreement? How long is the agreement for, and what does the royalty structure look like?
Sure. So what we've been allowed to disclose by Takeda is a couple things. One, this sublicense was executed back at the end of 2017. At that time, we received a $7.5 million upfront payment, and then announced that we'll receive an ongoing low single-digit royalty. So that's about the extent of what we can disclose, and we can't disclose the name of the third party, the sublicensee, who's actually selling the product that's generating the royalties. I will say that the royalty stream commenced last year. Again, we've reported the royalties on a quarterly basis. They were somewhat modest. How much of an impact COVID had on the product launch is a little bit difficult for us to say because we're somewhat removed from it. So we would certainly hope those royalties would ramp. But the royalty stream, will be ongoing for a number of additional years. And if PharmSynthèse is able to successfully get approval for the polycylated EPO product, EPOLONG as they call it there, that would also be a revenue and royalty generating product, although obviously the Russian market is not as big as the US market.
And going back to XCART, when do we expect to see some clinical data? And also you mentioned it will be an Eastern European trial. When do you think you will have US clinical trial initiated or data from that?
Right. So we really want to focus on the collaboration in Eastern Europe as a way to get the first in-human trial. Now, as I noted in our upcoming milestones, really, if we're looking at 21, it's more a question, I think, of starting the manufacturing process and moving toward a phase 1. I would not expect us to be in a phase 1 dosing trial by the end of this year, but I would expect that to happen in 2022. And then once we're able to, it's likely, at least on our current plan, that we would try to secure some initial data from that Eastern European collaboration prior to filing a US IND. So US IND would follow that. That could change, but that's the current expectation and plan.
Thank you so much for your time. That covers all my questions, Jeffrey. Thanks for your presentation and presence at our conference.
Great. Thank you, Ahu, to you and to all of your Noble colleagues. Really appreciate being here.